It's a great honor to be here. Thank you for the invitation, all the organization from the leadership and everyone around this meeting, making it so fantastic so far. I wouldn't have dreamed of this kind of reception. It's so beautiful and it's so interesting. We already have some discussions about science and spirituality. And I do enjoy the food so much. Thank you, all of you. So when Jaya Prakash asked me to participate here in this conference on science and spirituality, I said, oh, that sounds really interesting. And I want you to deliver a talk about epigenetics and spirituality. And I said, that is a challenge. I never even thought about it before. So it's been a challenge, and I decided to give a presentation about the basics of epigenetics. It's very basic, and then hypothetically how epigenetics might be involved in the feeling and the perception of spirituality. By no means, I'm not telling you the truth, I'm telling you the hypothesis and ideas that might be behind that could be scientifically explored further. So my talk will be coming more focused on inside the organism rather than going out. So I decided to give the talk the title, Why Epigenetics May Be Relevant for Explaining Spirituality. So I'd like to start with DNA. As you know, the DNA is the only known form of biological function. Besides virus and stuff, they also use RNA. And the amazing thing with DNA is that it's a lot to regulate in a very tight space, which is the cellular nucleus. And for humans and mammalians mostly, then it's about 2 meters of DNA in one cell. Human body approximately contains 50 trillion cells. This means 100 billion kilometers of DNA in one human being. The distance to the sun from Earth is only 150 million kilometers. One person's DNA equals 2.5 million turns around the Earth equator. And here's like a visualization of how much DNA. So the distance between the sun and Pluto represents about 15% of the distance of all the DNA that one human being has. So you can imagine that it's a lot to regulate, and it all has to fit in the cells, and it has to be, the cell has to be able to use it. And so the DNA doesn't exist as just a spread out molecule. It's rolled up around proteins that form so-called nucleosomes. There's histone proteins forming nucleosomes, and these nucleosome DNA is forming, rolled up and forming a tight structure, which here is represented by the 30 nanometer fiber. This fiber is further compacted into more and more complex structures. And to the right you see how it looks during cell division, the metaphase chromosome. And we have, as you know, 46 chromosomes in humans. So it's a lot of DNA, a lot to regulate. So then what is epigenetics? Well, epigenetics means above. It's Greek and means above or over. So it's over genetics, on top of genetics. It's the regulation of the genetic material that is not immediately dependent on the DNA sequence. And most of you know that DNA consists of nucleotides, adenine, thymine, cytosine, and guanine, and the order of that decides how proteins will look and function in a human organism. But only a part of our genome is actually coding for proteins. There's a lot of DNA, which just 20 years ago was called junk DNA, And we now know that this DNA is exceedingly important to regulate those maybe 20% of DNA coding for proteins. So all of the DNA, more or less, is important. And in one organism, virtually all cells have identical copies of our genomes. So at cell division, the DNA information, including the epigenetic information, how genes are regulated, are replicated to the new generation of cells, from the daughter cell, from the mother cell to the daughter cell. And I would like to define then the genetic component, the DNA, as the hardware of the organism, while the epigenetic is the software which can be changed. So the hardware basically is not changed unless there is mutations, but the software can be programmed and reprogrammed. And the most typical example of how epigenetic works is that starting with embryonic stem cells, which are basically open to negotiation what to be later on, they are first differentiated by epigenetic programming, software programming into the 3 germ layers, ectoderm, mesoderm, and endoderm. And the further you go into differentiation of specialized cells, it is followed and guided by epigenetic changes, programming of the DNA. And of course you can't have epigenetics without genetics, because the genetics actually codes for the proteins that are involved in the programming. So it's everything is involved in itself. So what is then the epigenetic regulation? We have something called epigenetic code, which defines genomic regions and domains. And the mostly studied epigenetic modification is methylation of DNA on cytosines, called methylcytosines. The DNA is also, as I mentioned before, it's rolled up against around proteins called histones. Histones also modified by epigenetic modifications. So all of this forms a structure in time and space. Space because there are different places, and time because it's depending on when it's done at different effects. So you can see epigenetic and epigenetic modifications as a barcode. Barcode for a cell type. It's unique for each cell type and defines and controls the memory and properties of a cell and an organism eventually. So then epigenetics forms something we call the epigenotype. And the epigenotype is exactly what I said before, that it consists of various modifications, DNA methylation, histone modifications, nowadays also non-coding RNAs, chromatin modifying proteins that affects how the structure of the DNA into chromatin is formed. This is epigenotype. And this is a product, I would say, from both the environment and the genotype. It all forms then eventually the phenotype of the organ, or the cell of the organism. Environment listed here, blood diet, toxicant, intrauterine environment, early life experience, smoking, and so on, we know can affect the phenotype, the organism directly, but also indirectly, depending on the genotype that we have. So we all have different, a little bit different DNA. We have polymorphisms that define us as unique individuals. We can have different abilities to, for instance, detoxify compounds and to handle immune in the immune system in different ways. So by changing epigenetic states in a bad way, we lose something that we call phenotypic plasticity, and this can result in disease and aging. And I mentioned earlier that there are genomic domains defined by epigenetic modifications and erosion of those domains, which basically reduce instability, it will cause, or at least can cause, cancer and other diseases as well. To illustrate a little bit how we are different, here's just one region of chromosome 9. It's 50 individuals, variable DNA methylation in this region that you see before you. These 50 individuals have, in this region, different methylation here. And what is interesting is that in this very particular example, here is a region where smoking responsive DNA methylation is 30-fold in width. And we call these regions variable methylated regions. So, one practical example of epigenetics in action. There is a very important factor for modification of DNA, which is the folate metabolism. The folate metabolism works as a methyl donor, so that vitamin B12, methionine, tetrahydrofolate makes adenosyl methionine, which with enzymes methylates cytosine to 5-methylcytosine, leaving S-adenosyl homocysteine, and then it goes back into the metabolism. So you can affect the pool of methyl donors by changing the diet. Here we have a practical example. It's called the agouti gene. Were performed with mice, inbred, very, very similar genetically mice, carrying a gene coding for the agouti, which is a protein regulating melanin in the hair follicles, but also regulating hunger and satiety and so on. And this gene in this mouse is regulated by a old viral sequence called the retrotransposon. And this is normally silenced by methylation, but if you feed the pregnant mice mother low folate diet, which would then lower the methyl donor pool, you can open up, activate this retrotransposon activating element so that the agouti gene is expressed everywhere. And this results not only in yellow mice, but also in fat mice that also are prone to get cancer. So this is a practical example of how epigenetic works. In the human context, scientists found that women during the Second World War, when the Nazis were isolating villages in the Netherlands, women starving in the first 6 months of pregnancy gave birth to small babies who grew up to be more prone to obesity, coronary disease, schizophrenia, and cancer. And actually this is an example of an intergenerational effect, but we also know now that there is something called the transgenerational effects, that also the grandchildren of these women are affected, most likely through epigenetic mechanisms. And I compiled some interesting things from epidemiological findings regarding intergenerational effects, which means one generation only. How various things can affect through probably epigenetic mechanisms. So preconceptional effects, diet, smoking, drugs, alcohol. You can see on the left side, on the right side, and they can have effect on preterm birth, chronic diseases, anatomical defects, and so on. In utero effects, maternal depression can cause neurobiological effects. Maternal diet, as I already mentioned, for mice, so effects on folate and vitamin C. So, there are effects on the next generation and even further generations through mechanisms that are potentially and most likely epigenetic, not genetic, so not mutation. And this is interesting scientifically and biologically, and might also be interesting from a spiritual point of view, because it seems that spirituality could be kind of running in the family. So, next part, monoamines. You know that norepinephrine, serotonin, dopamine, very popular to talk about today. They are the three key neurotransmitters, very important for, among other things, mentally, mode, and cognitive function, and also have other functions, of course. When the nerve signals coming to the synapse, the vesicles that you see in the presynapse there releasing by melting into the membrane, melting and releasing neurotransmitters into the synaptic cleft. This is then bound to receptors, and the nerve signal is propagating to the next kind of dendrite. Now the neurotransmitters is going back through transporters, and this is important to be reused. Here is a protein which is very important for that, the Vesicular Monoamine Transporter 2, or VMAT2. So what happens is that when, in this case, it's dopamine has done its action by binding to the receptor in the postsynaptic membrane, it goes back through the dopamine transporter into the cytoplasm of the presynapse. And then running around there in the cytoplasm, it has to be collected, and the VMAT2 is transporting those neurotransmitters back into the vesicles so that it can be reused. And this is important to have this balance. For instance, if you look here to the lower figure, which is the dopamine transporter level, if that is high, the cytosolic dopamine is high, and the extracellular dopamine is low. When the VMAT level is high, then the cytosolic dopamine is low. So the balance between these two functions, the transporter and the VMAT2, is very important to keep mentally and other functions in okay shape. So, now we're coming into some interesting things. There is an hypothesis proposed by Dean Hamer maybe 20 years ago, really, that there is a hereditary basis for the predisposition to spirituality and religiosity. If that's true, then he put up 5 different ways of looking at it. That spirituality can be quantified by psychometric measurements. 2, the underlying tendency to spirituality is partially heritable. 3, part of this heritability can be attributed to the gene VMAT that I just talked about, and which I have added here to VMAT gene regulation, which is going then back to epigenetics. 4, this gene acts by all 3 monoamine levels, which is important. And 5, spirituality provides an evolutionary advantage by providing individuals with an innate sense of optimism. And this is how evolutionary this has survived, maybe. This is the hypothesis. So, if this hypothesis is correct, there are several possibilities, and 2 of them, which I thought about, is that 1, the VMAT2 gene, and by all means, maybe many other genes, and variants of these genes predispose individuals to have spiritual and religious experiences. Or 2, that the VMAT2 gene, or others, and the variants enhance the perception of something real and greater than themselves, divine presence. So I call it sensitive antennas that exist in some individuals and not so much in others. So, I've talked about this. Epigenetics is the operating system of cells. Various kinds of impact by the environment, including, for instance, diet, can reprogram the genome, leading to phenotypic change. Gene-environment interactions involve, for instance, DNA information from early development to adulthood and over generations. The God hypothesis involves a VMAT gene variant that predisposes to spirituality. VMAT2, as other genes, is of course subject to epigenetic regulation. So, food for discussion. Might drug abuse affecting monoamine levels? We know that they do. Could that epigenetically perhaps alter genes in this regulatory system, including VMAT, that leads to propensity for spirituality and religiosity? Conversely, evidence suggests that religious faith and spirituality can play a significant role in the recovery process for individuals struggling with substance abuse. And a study has actually shown that 73% of substance abuse treatment in USA has a spirituality-based element. Another study found that those with stronger religious beliefs are as much as 8 times less likely to use illegal drugs and as much as 5 times less likely to binge drink. There could be many reasons for this, but there could also be biochemical reasons. Can genetic and epigenetic variations result in propensity for spirituality and religiosity? That's one question. Can early spiritual religious exposure, now we talk about the environment, trigger changes in gene regulation, leading to spiritual or religious experiences, that is, consequences from being raised in a spiritual family? These are things that one could discuss and continue to talk about. And I would like to end with the quote by Stephen Hawking: "The greatest enemy of knowledge is not ignorance, it's the illusion of knowledge." And I take that as, don't deny things that you don't know anything about. Continue always to seek knowledge. We will never get to the end of this quest, but don't think that you know everything, because you don't. Thank you.